Hey, Paul, welcome back.
Thank you. I guess we can jump right in.
We can jump right in. We're already recording.
Okay. Share screen. You need to make either allow share screen or make me host.
I will make you a host.
Awesome.
There you go. Okay.
Great. Thank you.
I'll put myself on mute and
away we go.
Go to it.
Okay. Okay, this is part 2 of my presentation on can universal common descent be tested by molecular data? And let's just pick up the thread directly from where we were yesterday. Here's our outline. Today we will be looking at points 4 and 5 of the presentation. Sorting complex bundles, the yes answer to the question, can universal common descent be tested? Yesterday we explored the no answer. And then concluding with part 5, looking at what I call the heart of the problem, Sober's paradox. So I'd like to point out before we get going that the molecular examples I'll be giving in the next hour could really be multiplied over the length of a semester. So in fact, I'm working on a book right now addressing that. The examples therefore represent a theme. They're something that was unexpected throughout really the second half of the 20th century until large-scale genomic sequencing became a practical technology. So I'm just giving you a sample of a very much larger body of data that have proved quite surprising to comparative biology. And the question to keep in mind throughout the next hour is, if universal common descent were false, how would we know it? It's a tricky question to answer, and it's actually full of interesting kind of puzzles. So let's go back to the genetic code and the prediction of its universality, which is where we were when we concluded yesterday. And I'll just recapitulate briefly. The universality of the code was actually predicted 3 years prior to its elucidation in a paper published in Science in 1963 by Hinegardner and Engleberg. And their argument was quite simple and very strong. They said, if there were a mutation in the code, it would affect every protein produced by that cell. Unlike mutation in a single gene, which would affect only the protein product of that particular gene, a mutation in the code, the translation table between the realm of nucleic acid and the realm of amino acids in protein assembly, a mutation to that semantics, if you will, would affect every protein produced by that organism. Therefore, they said that would be very unlikely to be favored by natural selection. Bottom line, the genetic code, once established, would therefore remain invariant. So there's a strong prediction based on cell biology on the one hand, when coupled with the theory of universal common descent on the other. So here's a cartoon showing the prediction as I discussed it yesterday. Parsimony would lead us to infer that if the last universal common ancestor existed and was the root of the 3 major domains of life here, bacteria, archaea, and eukarya, that it would have possessed a DNA genome, a genetic code, and other major players in cell biology. And like the conserved release factor prediction that we discussed yesterday, one would expect to see the code present throughout the 3 domains in universal form, because mutations to the code would be or were thought to be inevitably lethal. So there's the prediction. Now, we can lay it out this way. here's our prediction. Here are the three major domains. And here's a typical expression as one would find it in a textbook from Ridley, 1985. The code would be retained because any change in it would be disastrous. A single change would cause all the proteins to be built wrongly. No such body could live. Thus, we expect the genetic code to be universal if all species have descended from a single ancestor. So this is the prediction as you would have found it from the mid-1960s, really until quite recently. Place your bets. How did it do? How did the prediction do in the light of new data? Well, not so hot. So here is a cartoon from an article in the journal Current Biology showing some of the variants present in the three domains. And this is an incomplete selection. It's just giving you some of the diversity that's there. Commentators looking at this pattern more recently have said, we once thought the code would be universal and immutable. It is now known to contain many variations, including biased codon usage, codon reassignment, and ambiguous decoding and recoding. And actually, the NCBI maintains a web page where you can go look up some of these variant codes, which in fact is important to do if you're working, for instance, with a ciliated protozoan, like a tetrahymena or paramecium. Here, these three codes here. The ciliate codes vary in interesting ways from the so-called canonical or standard code. I won't call it the universal code anymore because, in fact, it's not universal. But it's important if you're working with a model system that uses a variant code to know that so that you can predict protein sequences correctly. If you take a DNA sequence that you've obtained and read it out through the so-called canonical or universal code, and your study group or your model system actually uses a variant code, you will incorrectly predict protein sequence or amino acid sequence because you're using the wrong code. And there's an interesting exchange online between Richard Dawkins and Craig Venter about this, where Dawkins says, well, you know, the genetic code is universal, and Venter interrupts him and he says, well, that's actually not true. I just finished an experiment with my group, Venter goes on, using mycoplasma, which has a variant code. And he says to Dawkins, your proteins wouldn't work in mycoplasma because of that, because of the variant code possessed by mycoplasma. So it's an interesting area to explore, and new variants are being found with some frequency. Now, a standard reply to this, to sort of save the prediction, is to say, well, it's nearly universal. This used to bother me only a little. Now it bothers me a lot. When someone says nearly universal, I regard that as a nonsense phrase. So here's a way to think about it. And you need to think carefully about this, because nearly universal actually is, I wouldn't call it an oxymoron necessarily. It is in fact literally a nonsense phrase. Universal as a designator is not a term that logically admits of degrees like height or size. So here we have, forget about the code, we have something that we're interested in and we say it's 99% universal. There are only 1% of variants that we found. Well, nearly universal applies here and here, but 99 and 90 are not equivalent values, right? Or here, again, you've got nearly, it's 80%. 99 does not equal 90, does not equal 80. We have slipped into the realm of nonsense. Universal does not mean occupying most of some space or distribution. That's exactly what it doesn't mean. So if someone says to you, the code is nearly universal, you need to stop them and say, that actually is a nonsense phrase, and don't use it. It's akin to approximately pregnant, almost alive, right? You need to think clearly about using a term like nearly universal and avoid it. So my little gripe session is over. Please don't say the code is nearly universal. You can say it's legitimate to say we once thought the code was universal. We now know that is no longer the case. Leave it there. Don't employ nonsense. All right. What happened to our prediction? This one. The universal common descent as a geometry or topology of relatedness rooted in LUCA, the last universal common ancestor. That topology, when coupled with the theory of functional invariance of the code, gives us this prediction. We should see a universal genetic code, whatever species we examine. Well, the prediction underwent a makeover. It was finessed, if you will. So in a very good book, which I actually have sitting right here somewhere, I don't know what I did with it. A very good book that I recommend to you, actually, by Matthew Cobb at the University of Manchester, which addresses the history and discovery of the code in great detail. It's really a wonderful treatment of this episode in molecular biology. When he comes to discuss the non-universality of the code, he says, look, this was completely unexpected, this discovery, and went against all the assumptions of all the researchers who had been studying the code. These discoveries showed that, strictly speaking, Jacques Monod was wrong. What is true for E. coli is not necessarily true for an elephant in all respects. So this prediction fails, and now someone has to pay. Someone, I'm sorry for personifying these theoretical notions, some part of our complex bundle of universal common descent plus auxiliary theory is responsible for this failed prediction. According to Modus Tollens, we've got to go back over there and find out where the problem is. You can personify nature. She's standing there in her police uniform, and she's got her ticket book in her hand, and she's writing out a penalty, a fine, for the failed prediction. And she's telling you something in your theoretical bundle is responsible for this, and that part has got to pay. It's responsible for your failed prediction. Something in there. Okay, now, what does Cobb do? He doesn't make universal common descent pay the fine. Rather, he passes the ticket to the auxiliary theory. And very next sentences in that passage I cited previously, although the genetic code is not strictly universal, this has not altered our view of the fundamental processes of evolution at all. There is no dispute that life as we know it evolved only once, and that we all descend from a population of cells that lived some 3.5 billion years ago, known as LUCA. So, prediction fails. The auxiliary theory pays the fine, and a universal common descent is insulated or protected from empirical challenge. So the metaphor I used yesterday was the queen in her throne room. She is sitting there enjoying a cup of tea, and the courtiers and the soldiers and everybody else is being hacked to pieces with the broadsword somewhere else in the castle. She is unmolested. Okay, universal common descent doesn't pay any observational fines. Now, watch what happens to the predictive strength of the theory if you do this. Okay, so let's change our functional invariance theory to something more tolerant of anomalies. We say, well, look, the code may vary. Who knew, right? Who knew? So our auxiliary theory is now the code may vary. Well, let's see what nature has to show us. If we put that in here, instead of functional invariance as our auxiliary theory, now the empirical content of our prediction is indeterminate. We don't know, right? The prediction is actually not telling us anything about nature. We just have to go out and look and see what we find. So the consequence for the predictive strength of universal common descent is it takes a serious hit. It's got indeterminate empirical content. A bunch of question marks is not very promising if you want a theory that's doing work for you. Again, the same problem I discussed yesterday, you end up with a theory that will be consistent with whatever you observe. And that means the theory is actually not telling you anything about the world. This is not a theory I would want to work with because it's not doing anything for me. And I may as well put it to one side and just go out and observe. Okay? So there is a consequence for doing this, for making the auxiliary theory pay the fine. Universal common descent ends up being robbed of its content. All right. Now, let's shift gears. I've shown you what has tended to be the practice of comparative biologists, that is, biologists who deal with these large-scale questions of the structure of the tree of life, the relationship of the three domains to each other, and so forth, who deal with what is the predictive content of universal common descent. The move that I just showed you of making the auxiliary theory pay has been viewed with increasing skepticism, if you will, by many evolutionary biologists, an increasing number, in fact, for reasons that have nothing to do really with the intelligent design debate. So let's look into that now in a little detail. So now we're going to look at the yes answer to our question, can universal common descent be tested? Step back, brief recapitulation. Right? The perfect generality of universal common descent, which is attractive for lots of reasons, but does come with a cost. Namely, it's hard to get a perfectly general theory to make predictions for you. In fact, it's impossible, I would say, in this case. It needs helper or auxiliary theories to make testable predictions. So this is how it looks. I went over this yesterday. When we conjoin universal common descent with other parts of our biological knowledge, then we can get observable predictions out. So this is the state where we want to be, because this, then we can get the cycle going of making predictions and finding out how to fix this bundle if it's not yielding predictions that actually obtain. All right. Now, what happens when a prediction fails? Well, if this is our complex bundle, we don't have to protect universal common descent. All right. And I would say this even if I wasn't an intelligent design theorist. If, sort of pretty much, I would say 50% of me is an evolutionary biologist. It's what I studied in graduate school. I can well imagine, for reasons, again, unrelated to intelligent design, seeing a failed prediction and saying, you know what? I actually know more about this, about my auxiliary theory, than I do about this. I'm going to make universal common descent pay the fine. All right? I'm going to hang on to this, and maybe this isn't true. So this is what has begun to happen in large-scale comparative biology since the advent of the genomics era. So in January 2009, so this is now, it's over 13 years ago. Boy, time goes by. In January 2009, 150th anniversary of the publication of The Origin of Species, New Scientist ran a cover story, Darwin was wrong, cutting down the tree of life. And this article was so controversial that many prominent evolutionary biologists were incensed. And they said, we'll never have anything to do with New Scientist magazine again. And I'm sure New Scientist was laughing all the way to the bank. But why? Why an article like this? Well, as it turns out, and you can go online and read the controversy that erupted, Graham Lawton, the science writer in the UK who wrote this cover story, he defended himself against these evolutionary biology critics. And he said, look, I don't care about creationism. Forget about all that, right? New Scientist is not pandering to the creationists. I'm simply pointing to developments within evolutionary theory in response to anomalous data that really no one expected, such as the work of Carl Woese, who died a few years ago, but who was one of probably, I would say, the most influential molecular biologists working on evolution in the second half of the 20th century. It was Woese's work on the RNAs that are present in the ribosome, the very large structural RNAs, structural and catalytic RNAs that are present in the ribosome, that gave us the three-domain geometry. And he had a number of very influential students. It's interesting to look at Wose's career because over the last 2 decades of his career, he became increasingly outspoken about his unhappiness with textbook evolutionary theory, including the single tree picture. And the notion that there was a last universal common ancestor as a discrete cell. And in this paper in the PNAS in 2002, he said, the time has come for biology to go beyond the doctrine of common descent. And this capital D here in doctrine, that's not a typo. That is his choice as author to indicate something that he regards as standing in the way of scientific understanding. A doctrine in this sense is a proposition or a set of propositions held for dogmatic reasons, very much along the lines of a theological doctrine, right? That's the sort of notion he's trying for here. And he says this is a doctrine that we need to go past. We need to reform and go past. This statement is made in the light of data that Wose could not fit to the single tree picture, such as his views on the nature of ribosomal structure and function. I wish I had time today to go into all the interesting details. Maybe in a future presentation I'll be able to do that. But bottom line, Wose said, look, there are molecular anomalies present here in these comparisons that we make among the major domains that cannot be reconciled with the classical single tree picture. And I believe this paper now is open access. So you can go and just download it and read it for yourself. But he's hardly alone. Wose was hardly alone. So Craig Venter, who, as you know or probably know, sequenced his own genome in a race in the late 90s into the early 2000s, a race with the US government to determine the sequence of the human genome. Following his work on the Human Genome Project, he went on a yacht and he sailed around the world for a couple of years. He and his crew sampling seawater, okay? You can sequence DNA right out of environmental samples. It's called metagenomics. And even if you can't grow the microorganisms that you sample in culture, you can still recover their DNA from these environmental samples and examine it. So he did that, took all these samples as he sailed around the world, and he found millions of genes that no one had ever seen before. So he comes back into harbor, so to speak, and at a round table in Connecticut in 2007, he said, I don't buy it. In light of the discoveries I've made about the amazing genetic diversity that we pulled right out of the environment, right, sailing around the world, I don't buy the single last universal common ancestor geometry. He says, I don't think there was a single ancestor. It's counterintuitive. There may have been thousands of them, and they are not necessarily so common. Meaning that if you do a Venn, I wish I have to use my hands, unfortunately, here. If you had a Venn diagram of the molecular hardware of these different groups, there would be some small region of overlap where there are shared genes and proteins. But much of the molecular hardware will be unique to the groups that one is looking at. That's what he means by not necessarily so common, and thousands of different ones. So his picture, which is, again, fully an evolutionary picture, this is not an intelligent design argument at all, is not of a single tree, but of a forest of trees arising from a prebiotic milieu where you have some shared elements, but many bits and pieces that are taxon specific. And what's going on here is, again, as I pointed out a moment ago, faced with anomalous observations, coming back to that complex bundle of universal common descent plus the rest of our biological knowledge, an increasing number of biologists are saying, hey, maybe universal common descent, at least as we classically understood it, going back to LUCA, is not true. They're making UCD pay the fine. So another example is Eugene Koonin, who is one of my favorite biologists working today. I think I read pretty much everything he publishes. It's so interesting. He is Russian by ancestry or by training. I believe he did his PhD at Moscow University. Came to the US as the Soviet Union was collapsing. Set up a lab at the NCBI. He's now a member of the National Academy, a very, very productive scientist. And in this paper in 2007, again, note the year, he says, I don't think there was a tree of life. So that's what TOL here in this quote stands for. He says it didn't exist. A single uninterrupted tree of life does not exist. Again, this is an open access paper, fascinating. I encourage you to go download it to read his arguments. Raoult is a French microbiologist at Marseille. Again, he is not an intelligent design advocate. He's an evolutionary biologist or microbiologist. In this paper with, I believe, one of his postdocs, he said, the tree of life concept presumes that all organisms are descended from a predecessor, ultimately LUCA, of course. He says, this is contrary to our current knowledge. Why? There are too many unique genes. Here he says 10 to 15% of the genes of each bacterial species. So he's looking now just at the domain bacteria, has no equivalent in any other. So what he's doing is as he says, as I look across the domain bacteria, there is far too much unique genetic information to be compressed back into a single common ancestor at the origin of that domain and ultimately going back to LUCA. So what's happening here, anomalous data, unexpected, he comes back over to his complex bundle and he says, there's no way you're going to get all those genes from a single common ancestor. That's got to go. That geometry has got to go. So if you read Koonin's work, one of the reasons that he is skeptical of the single tree picture is a phenomenon, again, really sort of a surprise that was given to us by whole genome sequencing. Something, it's got kind of a clunky name. It's called non-orthologous gene displacement, often abbreviated NOGD. And it's not my term. I don't like it, but there it is in the literature. But what it means is really interesting. So here we have a pathway. Let's say this is a metabolic pathway for some function X, and we've got 3 genes and their protein products in that pathway, A, B, and C. So that's in taxon 1, let's say, right? Then we move over to taxon 2, and we have what looks like the same pathway in terms of its product, function X, okay? But when we look at the pathway in detail, we find only gene B is present in both taxa, in both species. Instead of A in this species 2, we have gene K. Instead of C, we have gene Q. So what's happening is these are not orthologous, which is the technical term for they do not share molecular homology. They are not the same genes and protein products. And what's happened is, you can go either direction in this story, but from the last ancestor of these 2 groups, genes have been displaced by different genes, and their protein products have been displaced by other genes and proteins, even though these pathways are ending with the same function. The players in the pathway are not the same. So let me give you an example of this. Transfer RNA is the business end of the genetic code in all life on Earth. So here's a transfer RNA, and it's 2-dimensional representation with a familiar kind of cloverleaf pattern. And here is the anticodon. Down here, this is these 3 bases contact the messenger RNA strand. This is literally the business end of the genetic code right here. Up here on the CCA stem, this is where the amino acid attaches. So this codon, GCA literally, or ACG, excuse me, will tell the cell in the ribosome, insert, I forget which amino acid corresponds to that. I'm sorry, I should have put that on the slide, but there's an amino acid up here attached at the CCA stem that corresponds to this particular codon. Now, look at this position right here. It's known as G37. In many tRNAs, this is methylated. It's modified by a special enzyme. And actually, as you go around the clover leaf, if you look at a variety of different tRNAs, they have all kinds of special modifications that are really amazing. Again, I don't have time today to go into that. But an enzyme does this. It comes in here and methylates this base, all right? Now, if tRNAs are present in LUCA and the genetic code is present in LUCA, and you need to have functional tRNAs giving correct amino acid assignments on the, from DNA to messenger RNA into protein assembly, what would you predict about the enzyme responsible for this methylation? All right. Is it going to be homologous across the domains of life or not? Well, I think clearly the expectation is yes, it would be homologous, because this machinery, transfer RNA, and the enzymes that modify transfer RNA are so central to everything else in the cell. Protein assembly is not something you can mess around with, right? There are constraints on the possible variation. The prediction from universal common descent would be it's going to be that enzyme that does this job is going to be homologous across domains. So here is a cartoon representation of that enzyme. It's known as tRNA M1G37 methyltransferase. Kind of a long, kludgy name there, but that's the enzyme that does the job. Here is a cartoon representation of the enzyme from bacteria, from the domain bacteria. Run the story back to LUCA. We've got another domain here, archaea. Is it going to be the same or not? All right. It's not. These are not the same enzymes, okay? This is the bacterial enzyme here. Here is the archaeal enzyme. These are structurally not the same. So when this was discovered in 2004, the authors said, well, gosh, who knew? The invention of two distinct enzymes to catalyze that modification was unexpected. Ask yourself why, right? Pause and ask yourself why. It's unexpected because of the geometry of universal common descent, a very powerful, elegant geometry that tells you as you funnel back towards the ultimate last common ancestor, things will tend to become more and more similar. That is the expectation. So the fact that they're not in this case is unexpected. Given that the modification of that particular position, G37, is conserved in bacteria, eukarya, and archaea. It's all in all three domains. This demonstrates clearly the principle of non-orthologous gene displacement. Well, that's giving a name to a mystery. Nobody expected this, right? That these very different enzymes would be doing the same, playing the same functional role. And again, in more recent work, Kunin has said this pattern of non-orthologous gene displacement is everywhere throughout molecular biology. Move from system to system and you find enzymes, proteins doing the same job in a sense, but they're actually not the same molecular player when you examine them in detail. So I want to move on to Sober's paradox and just end this section with a kind of, I guess you could call it a thought experiment. It's really, if I had to locate the problem that we're dealing with, it is actually the empirical emptiness of common descent. That common descent doesn't really tell us anything. And we can see that when we look again at the prediction of a universal genetic code. So here's our domain, right? Remember, everything on Earth, all biology on Earth is somewhere within this domain. So we want to make a prediction about the code, and we say, well, somehow it evolves. There's a pathway, we don't know what it is, but the code gets up and running, and it's functioning in LUCA, almost by definition, right? As you go up into the domain, you find the genetic code present in all organisms there. You know, dolphins, pine trees, the bacteria in your large intestine, you, your dog, my cat over there, Boomba in the corner of my office. All of these very, very different living things share a genetic code. So you say, I by parsimony, I look at all of this variety, I go back down to LUCA, and I say the genetic code is there. It's functioning in our ultimate. It's so weird.
Yeah, same thing happened yesterday.
You know what? It happened at about the same time.
Oh.
So I think, is there any chance you're on a 40-minute?
No.
All right. Well,
are we not recording? Oh no, we're still recording. So.
Yeah, it says we're recording. So
I don't know what it is. It's sometimes like I have a friend at work who's it seems like her internet goes out at the same time. So just pick it up, and it seems well. Hopefully you don't have Comcast, or maybe contact your cable provider or your internet provider.
I do have Xfinity, but I do a lot of Zooms, and I'm not putting the blame anywhere. It's just kind of weird. As a computer programmer, I'm amazed like anything this complex works at all the time. So just keep going. All right.
Maybe biologists feel the same way. I don't know.
Oh gosh, that's for sure. Although life is amazingly robust when you think about all the ways,
all the error control systems, and stuff like
feedback loops. I'm telling you. So if somehow the code gets up and running, and we say
you're not sharing, just to let you know.
Oh, all right. Share screen. There we go. Share, and we are right here.
All right, I'll put myself back on mute then.
All right, so we've got the domain of common descent. All the biology on the planet is within that domain. We predict that the code is present in LUCA, and then we wanted to generalize from that about what we should expect to find as we move out and sample through the domain. And we say, well, it looks pretty important. It's functionally essential as far as we can tell, so we say the code cannot vary. Now, the important thing to realize is the strength of that auxiliary theory depends entirely on our sample size. And it may be the case that the code can vary, or maybe not, but our prediction relative to common descent does not come from common descent. Common descent is actually telling us nothing. What's telling us anything about our prediction is our actual sample, all right? So here, these little circles represent organisms or species we've looked at. This is what has the content, the sample, all right? It's the observations that give us the strength for this, or the strength or the lack of strength for our auxiliary theory. So this is going to depend entirely on this and what we know about the actual range of variation for codes. Common descent is doing nothing for us at this point. In fact, I would say it really can't, because you say, well, what built the domain of common descent? This mysterious word evolution, which can mean pretty much anything. You can vary or not vary. You can go extinct or not go extinct. Evolution as a theory in biology rules out effectively nothing. And the only way to give it content is to actually go out and look. But at that point, you've left the theory behind, and it's your observations. It's the size and depth of your sample that is the source of your knowledge. So our prediction using functional invariance can't be any stronger than the size of our sample. Common descent is sitting on the sidelines. I'm sorry, I'm wildly mixing metaphors, but the book that I'm working on this lays this out in detail with respect to the fossil record, developmental biology, molecular biology. I have been at this a long time, right? I defended at Chicago in 1997, and over the intervening now, 25 years, I've come to realize that the apparent strength of common descent is illusory. And it's actually only observation and sampling that tells us anything about biology. But I want to stop with this part, and I want to move on to Sober's paradox. And this will conclude the presentation today, and I think I will finish on time.